My name is Brad Coates. I'm a research geneticist at USGA-ARS in Ames, Iowa, and the lead of the Arthropod Genomics Research Group, which is a community of like-minded researchers within USGA-ARS, kind of working in genomics in general. So the AGSx is co-sponsored by the I5K and INRAE. So I'd like to, again, go ahead and thank everybody who's organized our sessions 1, 2, 3, and 4, and are going on through the first 4, through February on through May of this year. So these are listed right here, as well as in the website, which link will be provided very shortly. And kind of catch up on all the updates. Some of these are still a little bit in planning, so bear with us. But we have a very good sessions coming up in subsequently of this in April and May. So definitely stay tuned to updates on that on the website, as well as through our Slack channel and other outlets. So definitely always want to upfront thank many people who are involved in the background. This definitely does not go on by 1, 2, or 3 people. So we have definitely a lot of great administrative support, kind of led by Kevin Hackett, our national program leader, who is basically give the blessing and allowed us to do this in general, which gives us access to Glenn Haynes, which is our master of ceremonies. He is very good in the background running Zoom and getting all the registration set up for us. And then Childers, who does the Twitter feeds as well as the website. Rob Waterhouse, who manages the Slack channel very well for us. And Pia Olson, who will be working very hard in the background, doing the video recordings. And also Lindsay Perkin, who is doing our backup recording. So all this is made possible through a lot of work by them, and I thank them very much for that. So they're again alluding to several of the resources that we have available for you to access, that being the website, the link right here, and this will be dropped into the chat, so you don't need to go ahead and try to write these down real quick. There's free registration, which you've obviously registered since you were here, but you have others you know who would be interested in attending these or future sessions, please forward that to them. All these videos that the recordings you're going to see today are going to be posted on an I5K YouTube channel for future viewing. Those will be put out into posts and reminders that they are posted once that does occur following this session. And there is a Slack channel, there's a link there. And for first-time users, there is a requirement to sign up. So the link is here, and we'll put that into the chat, and probably Rob Waterhouse, if he is here with us, will probably drop that into the chat for us as well. And with that, I'm going to turn our sessions over to our 2 organizers, Alan and Sonia, and have them give the introductions.
Hello. I am Alain Vignal from the INRA French Research Institute in Agronomy in Toulouse, and Sonia from the same lab. So we organized this session and invited some very nice speakers here. So this session is about honeybee genomics, and honeybees is a fascinating subject to study. It's a managed insect species which has been managed since several millennia, I believe something like 7,000 years, for collecting honey, wax, and royal jelly. More recently, it has been turned to be very important for pollination services, especially if you have, fields, very large fields with single crops. And even more recently, it has been acting as a sentinel towards threats on biodiversity, biotic factors, but also abiotic factors like pesticides in the environment. It's also very interesting to study on the biological point of view, because it's a social insect in which there is division of labor. Workers, queens, males have different tasks to carry on. And finally, it's a haplodiploid species, polyandric. Means that several males will inseminate one queen, and which means that there are some very specific breeding schemes which will have to be designed for the species. So next slide, please. We have 3 speakers today who will give exciting talks on different aspects of honeybee genetics and genomics. First, Alex Pinto who is a professor at the School of Agriculture at the Polytechnic Institute of Bragança. I hope I'm saying it well, Portugal. We'll talk about hypotheses on the migration routes followed by honeybee populations across time and the influence on the structure of the Iberian honeybee populations present today. Then Garett Slater, PhD student at Purdue University, will give us some insights on the influence of polyandry on honeybee male traits. And finally, Jana Obšteter, who is a research associate at the Agricultural Institute of Slovenia, will talk to us about stochastic simulations of honeybee populations. Bert has already said, but you can ask questions during the talks in the Q&R box. Don't use the chat box. The Q&R is the place to ask your questions. Speakers may answer your questions during the talks, and we will have 20 minutes talks, and 10 minutes questions at the end of each talk using the chat. You can turn on subtitles if you use the subtitle button, which is on the bottom right of the screen, if it can help you in some way. So now we're going to go to the first talk by Alice Pinto. She's an associate professor at the Department of Environment and Agriculture and Natural Resources School of Agriculture at the Polytechnic Institute of Bragança, Portugal. She started working with honeybees during her PhD over 20 years ago on the genetic processes underlying honeybee Africanization in the USA, and has since then worked on the natural and the beekeeper-mediated processes shaping honeybee diversity patterns. So this includes the genetic basis of local adaptation and the conservation status of subspecies and populations. So she will now present her results on diversity and adaptation in Iberian honeybees. So your turn, Alice.
Good afternoon, ladies and gentlemen. I would like first to acknowledge the organizers for the opportunity to speak today. I started working with honeybees over 20 years ago during my PhD, and for my thesis I focused on the genetics of a feral population undergoing Africanization in South Texas. Today I will talk about the honeybee native to Iberia, the Apis mellifera iberiensis, which, like the Africanized bee, underwent Africanization at some point in its history. Iberia has been a refuge and therefore a place of divergence during periods of glacial isolation, but it has also been a contact zone during interglacial expansions, as reported for many plants and animals in this book. Due to its proximity to Africa, especially in the Strait of Gibraltar, Iberia has also been a bridge into Europe for the more fragile organisms, such as the honeybee. Altogether, these features make Iberia a very interesting place to study honeybees. As you probably know, the honeybee is native to Africa and Eurasia, where it evolved into at least 31 subspecies. This large amount of variation was grouped in the seminal work of Rutner into 4 lineages: the O in the Middle East and Western Asia, the A in Africa, the C in Southeastern Europe, and M in Western and Northern Europe, and now all the way to Western Asia after the recent discovery of a new subspecies in Western China. These 4 lineages were defined by Rutner from morphology. A 5th lineage called Y was later inferred from mitochondrial DNA data on Apis mellifera yemenitica sampled in Eastern Africa. This was the state of the art until the recent publication of Dogantzis and colleagues who identified from whole genome data 2 additional lineages, the L and U, both in Africa. In this same work, Dogantzis adds to a long-standing debate by providing new evidence regarding the origin and radiation of Apis mellifera, which has implications for the Iberian honeybee evolutionary history, as we will see today. The early work of Rutner supported an origin of the honeybee in northeastern Africa, followed by one expansion route into Eastern Europe through the Mediterranean, giving rise to C lineage, another one into Africa, giving rise to A lineage, and from Africa into Southern Europe through the Strait of Gibraltar, giving rise to the M lineage. Later on, Garner and colleagues proposed a very different scenario from mitochondrial DNA. This involved an origin in the Middle East, followed by 3 expansion waves, leading to African and European lineages, with lineage M colonizing Europe from a northern route. More recently, Whitfield used genome-wide SNPs to propose a radiation scenario similar to that of Rutner, including the origin of the M lineage in Africa. The major difference was the location of the center of origin in sub-Saharan Africa instead of northeastern Africa, as opposed to Rutner's scenario. The most recent proposal of Dogantzis places the origin back into Asia with colonization routes similar to those proposed earlier from mitochondrial DNA. The colonization of the M lineage via Gibraltar would explain the morphological and also the allozyme clines that run from North Africa to France, with Iberia harboring intermediate phenotypes, as shown here for the allozyme MDH. This pattern led early studies to propose a mechanism of primary intergradation for the origin of the Iberian honeybee. The problem is that this hypothesis is not compatible with the mitochondrial DNA patterns found in Iberia. Contrary to the gradual cline detected by MDH, mitochondrial DNA entails a sharp transition between the 2 lineages occurring in Iberia, with the Western European occupying the northeastern half and the African occupying the southwestern half. Because these 2 lineages are highly divergent, the hypothesis that Western European haplotypes derive from African haplotypes through accumulation of mutation is untenable. Therefore, maternal variation does not support the primary intergradation mechanism for the origin of the Iberian honeybee, as proposed earlier from morphology and allozymes, being instead more compatible with a secondary contact mechanism. This leads to rejection of the origin of the M lineage and therefore of the Iberian bee in Africa, leaving the Garnery/Dogantzis hypothesis as the most likely. But then the question is, how did the African mitochondria arrive into Iberia? In this publication, Frank and colleagues explain the presence of African mitotypes in Iberia as a result of importations by the Arabs during the occupation between the 8th and the 15th century. However, the hypothesis of recent historical human-mediated colonization raises at least 2 problems. The first problem is that the diversity inferred from the popular intergenic region of the mitochondrial DNA is much higher in Iberia than in North Africa, which is unexpected considering a putative importation bottleneck. Besides, the most frequent African sublineage A2 in North Africa is relatively rare in Iberia. On the other hand, haplotypes of African sublineage A3 ancestry are very common in the Atlantic coast of Iberia and virtually inexistent in North Africa. The second problem is that in the study of rank, microsatellites failed to capture any structure or traces of African genes in Iberian populations. Instead of showing a gradual climb, like other nuclear markers, microsatellites retrieved a sharp break between Iberian and Northern African populations. Frank and colleagues speculated that selection was the most likely cause shaping the maternal cline, and that microsatellites could not capture the cline due to their neutral nature. Selection has been repeatedly invoked to explain the latitudinal clines captured by morphology, allozymes, and mitochondrial DNA. Yet, this hypothesis had never been addressed until the large-scale study that my group ran in Iberia. So, in an attempt to further understand the Iberian honeybee history, we started by building a comprehensive sample collection covering the entire Iberian honeybee range. To that end, we sampled over 700 colonies from 3 north-south transects as shown in this map. This comprehensive sample collection was then genotyped using over 380 SNPs distributed across the 16 honeybee chromosomes. We then conducted genome-wide scans for selection signatures using 4 different FST-based outlier methods for cross-validation purposes. We also used the program MATSAM to look for associations between genetic and environmental data for the identification of factors that might be correlated or act as selective pressures. The outlier SNPs detected by the FST-based methods are shown in these figures as the red dots. We found 34 SNPs that were cross-validated by at least 3 outlier methods. Of these, 15 were strongly associated with 1 or more environmental variables, and precipitation was the most frequent associated variable. Of the 34 SNPs, 32 were located in or near protein coding genes involved in a diverse array of putative functions, and 27 were mapped to genes, with only 9 of them laying in exons. The most compelling evidence for selection was obtained for a subset of SNPs mapped to different genes putatively involved in the same function. Just to give an example, 5 outlier SNPs were mapped to 5 genes implicated in vision, which is a very important function in foraging and mating behaviors. Interestingly, 3 of these 5 SNPs were strongly associated to insolation, which could be the causal selection pressure. The frequency distribution of one of these 3 SNPs is represented in this map, which clearly shows declinal pattern reported for other markers before. We then resequenced the whole genomes of a subset of 87 males chosen across the Iberian transects and then employed a battery of analytical tools in search for selection footprints across the entire genome. We used again conceptually different selection methods, and this time we performed, in addition, in silico protein modeling for functional selective SNPs. We found over 600 outlier SNPs detected at least by 2 selection methods, and many of these were associated with precipitation longitude and latitude. Genomic signatures of local adaptation could be identified in 140 genes, many of which are putatively implicated in fitness-related functions such as reproduction, immunity, olfaction, etc. Most outlier SNPs fell outside of exons. Within the SNPs mapped to exons, 41 were non-synonymous. Of these, only 7 could be analyzed by in silico protein modeling. These 7 SNPs are likely direct targets of selection, as they lead to amino acid replacement in functionally important sites of their respective proteins, as illustrated and in these figures. We show the predicted proteins for 3 genes that harbor non-synonymous SNPs located nearby important places in the protein. The maps below the protein structures show the geographic patterns of the amino acid variants produced by the SNPs. As before, we see structure in Iberia, this time using amino acids, with the map on the right side showing a clear northeastern southwestern gradient. So, selection does seem to play a role in shaping Iberian honeybee diversity. After filtering out the SNPs under selection, we asked 2 questions using the neutral dataset. First, we asked whether the remaining neutral SNPs are able to capture declinal signal in Iberia. The second question was how concordant are the patterns of neutral and mitochondrial DNA variation. These questions were addressed using spatial PCR, cluster and geographic Klein analysis. This figure shows the pattern inferred from neutral SNPs using spatial PCA analysis. Contrary to microsatellites, which were unable to capture the structure, neutral SNPs clearly capture the northeastern southwestern Klein that we saw for mitochondrial DNA. And the SNP pattern is strongly correlated with mitochondrial DNA pattern, as shown here, with a correlation over 0.81. So this cytonuclear pattern strongly suggests the presence of a secondary contact zone in Iberia, which was then confirmed by geographic Klein modeling. According to Klein theory, if variation has originated from secondary contact, then we expect neutral SNPs to detect clinal patterns and multiple coincident clines. In contrast, coincident neutral clines are not expected if variation originated via primary intergradation. Secondly, if a maternal cline formed by two highly divergent lineages is observed, and this cline is paralleled by nuclear DNA variation, then there is strong support for secondary contact. We tested this hypothesis on the SNP and mitochondrial datasets by cline shape modeling using the AGSx software. This slide shows the results of the cline shape modeling for 7 neutral SNPs, for mitochondrial DNA, for all neutral SNPs, and for the total SNP dataset, including SNPs under selection. This analysis revealed the presence of multiple neutral clines which share the center at kilometer 665, marked by these red lines. Furthermore, the maternal cline is coincidental and shares the center with nuclear clines, providing strong support for the presence of a secondary contact zone in Iberia. To conclude, there is no support for a mechanism of primary intergradation for the origin of the Iberian honeybee. There is strong support for the presence of a secondary contact zone in Iberia, a signal that microsatellites failed to detect. There is also evidence for selection shaping diversity patterns in the Iberian honeybee. These evolutionary and demographic signatures in the Iberian honeybee genome are still evident despite intensive beekeeper-mediated processes involving annual large-scale migrations within Iberia and queen breeding and trading. Well, I believe that we resolved part of the intriguing patterns exhibited by Iberian populations, yet there are still many pending questions. Here I listed some of them, which I hope my new PhD student is able to answer by integrating whole genome data from Iberia and Africa. The results I presented here were part of the PhD dissertations of my former student Júlio Chaves Galarza and Dora Henriques, and the questions that remain unanswered. I hope that Carlos, my new PhD student, will be able to address them. This research was published in several papers shown here that you might want to take a look. Several colleagues and funding agencies contributed to this research. That's all for today, and thank you very much for your attention.
Okay.
Okay, well, thank you very much for a very nice talk. I don't know if you've seen the one, there's just one question in the Q&A for the moment, which is how will honeybee pan-genome sequencing efforts with high-quality assemblies support your analysis? I suppose this means combining the sequence of several genomes together to have the alternative alleles for each position.
Alice, maybe
Hello?
you can answer by speaking rather than writing.
Hello.
Yeah,
Shall
thank you
I-
very much for being connected.
I was trying to write the answer, but if I can speak, it's easier and faster. I don't know. Shall I turn on my camera?
Yeah, sure.
Yes, sure.
Hello.
Hello.
Hi.
Good afternoon, good morning, and good afternoon to everybody. Thank you for being there. About the whole genomes, most of the data that I presented today come from the SNPs, not from the whole genome sequencing, only the selective SNPs. Part of the SNPs under selection. So we have the whole genomes 87 for the Iberian honeybee and many more for Africa. And our next step is going to be to look at these questions that I posted using these whole genomes. This will be part of the PhD of Carlos. So I hope, I think they will support. I mean, we did some analysis with a mixture, and we found the declines, like we did find for the small set of SNPs and for mitochondrial DNA. So they obviously they captured the declines. Well,
Okay.
I don't know if I answer your question.
Perhaps the question was also for other type of markers such as large insertions and deletions, which may be able to be genotyped in the near future using pangenomes. Anyhow.
Maybe we have one other question that is asked on our side. Sorry, it's not in the Q&A. There's another one in the Q&A, sorry. Okay, let's answer this one. So can this research be used to increase bee diversity and bee health worldwide?
I don't know. Well, of course, if you can increase diversity by migration, by gene flow, eventually it's a way to artificially, by importing bees from different ranges, one is able to increase diversity of local populations, and diversity is linked to health. So hypothetically, yes. And there is another question from
okay, wait, I have a related question to this one, I think, and also Jana asked it in a panelist discussion. So what is the current status of commercial and imported bees in Iberia, and can it explain somehow what you see in your data? Like, do you have some constraints in some regions, for example, that could explain that you have this decline?
Yeah, it's. I was really surprised with the results that we got, so clear. I mean, the signature of that ancient event, it is still so printed in the genomes. Although there is, beekeepers, they move colonies along every year in the Iberian Peninsula. There are 3 million colonies in Iberia, and 1 million colonies are moved annually. The beekeepers go after the nectar flows, and it's amazing that we still see the strong structure so clear. So I think this is possible because maybe there is selection going on that keeps the structure, but it is also it may be due to the fact that the mating occurs locally before colonies are moved. And queen trading, there has been importation of queens of C lineage ancestry, these commercial favorite queens. But we didn't see signals of introgressive hybridization. I was also surprised. We saw in these 700 colonies, one single colony in Portugal, actually, that still had the mitochondrial DNA of C lineage ancestry, but at nuclear level was exactly the same as the other Iberian bees. So. It's really, I was surprised and pleased to see that the Iberian bee is still very well preserved.
Okay, thank you very
So,
much.
yeah, I think we have a related question, Martin Hasselman. How frequent is colony transportation in Portugal and Spain? I suppose that one difference it could be between Europe, it's probably true in France also, that colonies do get transported, but perhaps not at such long distance as what I hear in North America, in which they go from one end of the country to the other. For instance, there is some migrations, but they do remain local. So perhaps the ones from southern Spain don't go way up north completely.
They go. I mean, the scale is not comparable with the scale of the US. But at the Iberian scale, the movements are very large, and they go from the more African component to the northeastern. So I expected to see a less strong structure like we saw.
No, it's impressive.
Because the movements are large, are like they can move the colonies over 1,000 kilometers away from home.
There's a question about going back in time from Rob Waterhouse. Do you have any plans of sequencing museum samples to go back in time and investigate ancestral diversity?
That would be nice to do that. Melanie is here and she did. I know that it's challenging to work with the museum samples, but yeah, indeed it will be nice to see what was the ancestry a few decades ago. But yeah, our plan now is to use samples from real populations. We have a project where we are going to sequence many hundreds of genomes from North Africa and from Middle East, actually from other different lineages and subspecies, which we hope that they help us understand the history. Because I believe that the colonization of Iberia, it happened not as suggested by the group of Garneri, like during a few hundred years ago, during the occupation of Iberia by the Arabs. I think it was much more. It is much more ancient than that, I wonder how much we see that in mitochondrial DNA. We see a break also in the genomes, a break between Iberia and North Africa, but I'm sure that there are still segments of the genome of Iberian bees that originate from these ancient colonizations. So we want to test those hypotheses.
Meaning you would like to look at haplotypes rather than global genomes?
Yes, that's why, I mean, we use the drones. We are going to sequence drones.
Yeah, very sense.
Exactly, to be able to have that power. I think the sequencing the drones is much more powerful than the workers, because we have the haplotypes. And yeah, so the 87 that we sequenced already, they are drones.
Well, you said to favor, like the recent paper of Dogantis, the hypothesis of Garneri et al. With the originate in
Asia,
Middle East. But couldn't it be that it did originate in Africa? There was the first migration, thousands, 10,000, I don't know, a long time ago, and then, as you mentioned, a more recent, the claim could be due to a more recent migration through the same route, as you mentioned, by the Arab invasion in the 15th century.
I don't believe in this historical recent colonization by humans that would explain what we see. I don't know if this is, I'm not sure if I understood the question. So I don't believe in that hypothesis first, because when we look at that intergenic region, the composition is completely different in Iberia from the North Africa, and we have much more diversity, which is kind of puzzling. And why, at the same time, why would the Arabs bring so many colonies? There were already bees here. I know that we humans tend to move things, we introduce so many colonies in the Americas, right? But doesn't make much sense. But the genetic data tells us that it's not, I don't think the genetic data support the hypothesis of human-mediated recent importations. Well, whether the origin is in Africa or in Asia, I believe more in the origin in Asia, as explained in the paper, the recent paper of Dogansis. The big difference between the paper of Dogansis and the paper of Garneri, although the scenarios are very similar, is about time, because in the Garneri paper, the lineages split about 1 million years ago, whereas in the Dogansis is about 6 million years ago. And that makes more sense to me also, but we will have to investigate for the case of the Iberian honeybees and when that colonization happened. I don't think it happened recently.
Okay. Well, I think you're right. There's a question about why don't we see any African ancestry in Iberia if there was a secondary contact? I suppose this refers more to the microsatellites than the SNP markers. Yeah. Why do you think we don't see any African ancestry in Iberia if there was a secondary contact? But I think it was mostly...
We see for mitochondrial DNA, because there are two lineages, African and mitochondria. And that pattern is paralleled. The northeastern, southwestern pattern is paralleled by the nuclear DNA. The thing is that it's the divergence between the Iberian nuclear DNA is high relative to the North African bees. But that component, I don't know if I can tell that component is African derived.
Yeah.
No, I believe that there were many, these groups of bees were in glacial refuges, and there was contact between the interglacial periods during expansions. I think I don't know how many times this occurred and for how long. But mitochondrial DNA, it's very clear in showing that the African bees were invaded Iberia.
Okay, and so the last question that we have...
Let me just say something, because I was thinking initially Rutner, when it came up with the 4 lineages, he placed in the Western, in the M lineage group, he placed the Apis mellifera, Apis mellifera iberiensis, and the Northern African Sahariensis and Intermissa. So they do share a common ancestry, and the early works also suggested that, even with morphology.
Okay,
sorry.
No, that's all right. That's great. Thank you for answering all these questions. So the last question that we have in the Q&A is about whether you used exclusion criteria to make sure that your 700 sampled colonies would not be suffering too much from mixing of genetic backgrounds due by the beekeepers' practices.
Okay, when we asked, we avoided sampling from same beekeepers, so to avoid that they will be related. And we sampled apiaries that were many kilometers away from each other. I cannot remember exactly when this was, some time ago, but trying to avoid this shared, recent shared ancestry.
Yeah,
so
so you try by your sampling to control a bit for this. Okay.
Yeah.
Okay, great.
Well,
Thank you very much, Alice, for answering all these questions. Thanks everyone that also asked questions. And you had a lot of comments on the Q&A saying a great talk. Thanks so much for the really nice presentation. So thanks again for that.
and
Thank you very much for having me. And
It was great.
Great talk. It was very interesting. It's fascinating what's going on in Iberia with honeybees. And if people still want to ask questions, I think they could still go on asking questions in the Q&A, which you can answer later. And people could still go to the Slack channel and ask questions. The discussion can go on for
as long as you want.
As long as we want. So now we're going to go on to something slightly different. Garett Slater is a PhD student at Purdue University. But he has 12 years of beekeeping experience, including 8 working for a large commercial beekeeper. So he has much more experience in beekeeping than I have. He also worked with the Bee Informed Partnership at the University of Minnesota with 30 commercial beekeepers. His current work focuses on applying modern genetic tools such as genomics to honeybee breeding. And his topic he's interested at the moment is the influence of polyandry on the genetic variation in male honeybees. So, Garett, it's your turn.
Hello, my name is Garett, and I'm a PhD student at Purdue University. And today I'm talking about polyandry and how polyandry can shape genetic variation in male honeybees. But before I talk about that, I sort of want to talk about my general goals of my thesis. And so I really want to understand the genes and mechanisms that shape variation, both phenotypically and genetically, for male honeybees. And from an applied perspective, this is an important question. Currently, commercial operations are finding that queens are failing or unable to reproduce after 6 months. And this queen really should be properly reproducing for 2 to 3 years. And so while much research and energy has been focused on the queen part of things, we are increasingly discovering that the males actually may play a more important role for queen failure than originally expected. And so some of these queen failures are actually linked to poor sperm quality or poor male quality. And so it seems like there's quite a bit of variation in males that could be contributing to queen failure. And so I really want to understand some of these mechanisms that are shaping variation in male reproductive traits and how this could be contributing to queen failure rates. Now, I don't have time to talk about my entire thesis, but I want to talk about one specific mechanism that could be shaping variation in male honeybees, and that's polyandry. So honeybees are really weird because they've evolved polyandry independently of other closely related bees. And so polyandry just basically means females are mating with multiple males. And in the case of honeybees, these queens or females are mating with 14 to 15 males during these multiple mating flights. Now, if you compare that to closely related bees such as Tetragonula, Bombus, Euglassa, and Megachile, these females are mating with 1, maybe 2 males. Now, if you think about from a male perspective, this changes sort of the fitness from this sort of male-to-male competition for menageous bees, where in order for a male to be successful, these have to outcompete other males for female access. But once they successfully mate with a female, then from the sperm perspective, it's less important. But for honeybee males or polyandrous males, these males must not only compete against other males for female access, but they also must compete with sperm from other males to not only get stored and utilized during fertilization, which could be 2 to 3 years after insemination. And so polyandry really introduces this new concept of post-copulatory sexual selection or sperm competition. And just to sort of visualize sort of the impact of polyandry on some of these sperm competition or sperm-related traits, we worked with this collaborator, Dr. Amy Dapper, at Mississippi State. And she developed this model to help us kind of understand sort of how does polyandry impact the evolution of maybe some of these sperm competition alleles. And so this gray line shows a sperm competition allele that arises in a species that's monandrous. And you can see that this allele is really maintained at low frequencies. It really doesn't reach above 25%, even after 500 generations. Whereas if this allele rises in a polyandrous species, such as honeybees, it spreads and quicks at an extremely high rate. And so you can see that with the introduction of polyandry, that the evolutionary rate is significantly elevated. And so this could be playing a role in the divergence and evolutionary rate of some of these genes associated with sperm. So polyandry is also expected to intensify sexual conflict. And sexual conflict really means that females and males have these two phenotypes that are determined by the same genetics. And in order to optimize or maximize these phenotypes or traits, they have to compete over these similar genetics. And this creates what is known as sexual conflict. And so polyandry is expected to sort of intensify sexual conflict. And just to sort of demonstrate this, I want you to look at this graphic. And so for monandrous species, sperm competition trait, maybe testes, is sort of less important for fitness. And so let's say females have ovaries, males have testes, and they're determined by similar genetics. And so because sperm competition is less important for maximizing fitness for males, there isn't really sort of sexual conflict over these similar genes because males are more focused on sort of this male-to-male competition. However, with the introduction of polyandry, these sperm competition traits become more important. So for example, males that have larger testes that produce more sperm may have a fitness advantage over males that have smaller testes and produce less amount of sperm. And so that really shifts this fitness optimum to these extreme points. And so now these genes that were previously not under conflict between males and females now are, because in order to maximize fitness from both the male and female perspective, they're fighting over these shared genes. And so in order for males to really maximize these traits or get these traits that maximize their fitness associated with sperm competition, they have to overcome this sexual conflict. And they can do that in two major ways. First, they can just modify pre-existing mechanisms. And so in order to overcome this sexual conflict, they can maybe change these expression patterns, so have higher expression of some of these genes when they're expressed in the males, or maybe change expression through alternative splicing. Second, they can say, okay, well, we have these genes that we're kind of fighting over. Let's just evolve entirely new mechanisms or new genes, and then we can just overcome this sexual conflict altogether. And so for the duration of the talk, I'll be talking a little bit about how males may overcome this sexual conflict in order to maximize their fitness. And so just kind of summarize the two major talking points I'm going to cover. First, I'm going to look at how polyandry impacts divergence and evolutionary rate. So kind of our model predicts that polyandry should elevate evolutionary rate of some genes associated with sperm competition. And then two, how does polyandry deal with sexual conflict? Does polyandry increase sexual conflict? And if so, do males just modify pre-existing mechanisms to overcome conflict? Or do males sort of resolve conflict by evolving entirely new mechanisms? And so those would be the two major talking points throughout my talk. So to answer this question, honeybees provide a really cool model. Like I said, they're really funky because they've evolved polyandry independently of other bees. But this also provides a really cool model to test some of the hypotheses I have. And so in order to test some of these questions I have with the impact of polyandry on some of these male traits and variation, I have this phylogeny of bees that include honeybees, which are highly polyandrous, to monandrous bees, which includes Tetragonula, Bombus, and Euglossa. And so these 4 bee species are all corbiculate bees. And Megachile rotundata is not a corbiculate bee, but it's also monandrous. And this is sort of the outgroup I'm using in my phylogeny. And so I collected, with some help with several collaborators, males and females from these 5 different colonies. And I dissected brain and reproductive tissues from both males and females. And so this not only provides a comparison between monandrous and polyandrous groups, but also between different types of tissue, such as somatic tissue and reproductive tissue. I did RNA-seq analysis on these different tissues from these different bee species and did bias expression analysis. And so at the end, I'll be talking a little bit about these different gene sets. And so I have gene sets that are specific for male brain and female brain, testes bias versus ovary bias, and I have these for these different bee species. And so this bias expression analysis enabled me to produce these different gene sets. And then once I have these different gene sets from these different bee species that differed in these mating systems, I was able to look at divergence, selection, and do some evolutionary rate analysis. And so what's really cool about honeybees is they really provide me sort of a way to look at the impact of polyandry on some of these sperm competition traits. Okay, first I was interested in how polyandry impacts evolutionary rate. And to go kind of back to this graphic that we collaborated with Dr. Amy Dapper at Mississippi State, we predict that if a sperm competition allele were to arise in a menandrous bee species, that it would be maintained at very low frequencies. It would never really reach above 25%, even after 500 generations. Whereas in a polyandrous species, allele will spread and fix rather rapidly. And so you can see that we would predict that polyandry would sort of elevate the evolutionary rate and divergence of some of these traits related to sperm competition. To answer this question, we looked at divergence between honeybee populations. And so we compared Apis mellifera scutellata in Africa to Apis mellifera serrana in Europe. And if polyandry was sort of elevated in evolutionary rate and divergence, we'd expect higher sort of mutational differences between these populations. And so to test this hypothesis, we looked at DOS, which is a measurement of non-synonymous divergence between these two different populations. And essentially what it does, it looks at non-synonymous mutation differences between these populations and whether they significantly different. And like I said, non-synonymous mutations can affect sort of a trait. And so if evolutionary rate was sort of elevated, we would expect that some of these non-synonymous mutations would increase and spread rather rapidly between these populations. And so to look at that question, we compared seminal fluids, which is a really important group of proteins for sperm quality and sperm competition, and they're expressed exclusively in males. And so we see that there is a higher non-synonymous divergence or non-synonymous mutation differences between these two distinct populations. And so this shows that if a mutation does arise and has some kind of fitness benefit, that it can spread and fix rather rapidly within a population and cause some of these population differences. But polyandry is also expected to promote the divergence between species. And so we would expect that that for genes associated with sperm competition, that polyandry would promote higher divergence in these specific sort of gene classes, and specifically testes bias genes. And so we looked at DNDS and whether polyandry in fact elevated DNDS from monandry to this polyandry transition. And so in order to answer this question, we have these sort of two different comparisons. We have a polyandry comparison and a monandry comparison. And so this polyandry comparison is just really looking at the change in DNDS from our outgroup, Megachile rotundata, to our newly evolved polyandry group, which is honeybees. And so this is this transition from monandry to polyandry. And we compared that to sort of our monandry comparison, which is our transition from monandry, our outgroup Megachile, to monandry tetragonula. And if polyandry was in fact sort of promoting this or increasing this divergence, we'd expect a significant difference in the polyandry comparison versus the monandry comparison. And so we have our four different gene groups: female brain, male brain, ovary, and testes. And really to summarize this, we see that for the brain and ovary between the polyandry and monandry comparison, there is no significant difference. But what's really cool finding was that for the testes bias genes, we see this significant increase in DNDS divergence. And so this really indicates that when we go from monandry to sort of polyandry, that this does seem to elevate divergence, specifically in testes bias genes. And some of these genes are likely associated with sperm competition or sperm-related traits. And so what we really find is that polyandry not only promotes sort of these differences among honeybee populations, but sort of rapid divergence of genes associated with testes and possibly sperm competition. Okay, question number 2 is, how does polyandry deal with sexual conflict? And so like I talked a little bit about earlier, was that sort of these traits associated with sperm competition aren't under sexual conflict because males that section isn't favoring males with traits associated with sperm competition. Fitness is favoring males that have this male-to-male competition. However, once polyandry evolves, selection is now favoring males that have some of these traits associated with sperm competition. And so now males and females are fighting over some similar genes in order to maximize fitness for males. And so our expectation is that once polyandry evolved, these genes are now under sexual conflict. And in order to optimize some of these traits, males must overcome this sexual conflict by kind of resolving it in some form. And so I guess our first question was, does polyandry in fact promote sexual conflict? And we did find that polyandry does really promote sexual conflict, especially for genes associated with testes bias. And so 54% of genes that were testes biased in all the menandrous bees, and so these genes were consistently testes biased for every single bee species, became unbiased or were now under sexual conflict. And so 54% is an extremely high proportion. In comparison, only 1.6% of genes became unbiased after being overbiased in the other gene sets. And so this really does indicate that polyandry does in fact promote sexual conflict, and an extremely high rate for testes biased genes. Okay, we see that polyandry does promote and accelerate sexual conflict, but in order for males to kind of optimize their fitness, they have to overcome this conflict one way or form. And so one way to do that is really just to modify pre-existing mechanisms. And so one way to do that is to simply change expression. And so instead of really modifying these genes can be expressed differently based on whether they're expressed in a male versus female. And so in order to see if polyandry changes the expression pattern, which could possibly resolve this conflict, we once again did our polyandry versus menandry comparison. So going for our polyandry comparison, going from our outgroup Megachile to our polyandrous honeybees. For our menandry comparison, going from our outgroup Megachile to our other menandrous species, Tetragonula. And then we looked at the ratio of expression between females and males. And so if polyandry was changing expression patterns between sexes, this ratio would be higher between males and females. And that's really what we see across all of our different gene sets. For these female biased gene sets, ovary and female brain, this ratio is dramatically increased, nearly 5-fold higher for the polyandry comparison versus the menandry comparison. For the male-specific gene sets, we see a similar trend, but it's not nearly as extreme. But we do see differences in female versus male-biased expression. And so expression may be one form of conflict resolution. Number 2 is alternative splicing. And so we have a DNA sequence, and we have these colored exonic regions, and these black are the intronic regions. And in order to produce a transcript, these exonic regions are removed and spliced together. And based on their splicing, they can produce 2 different phenotypes. And so from this transcript number 1, you can see that all 3 of these color regions are sort of spliced together. And so this would be 100% efficiency. And from PSI, this would be a 100. Whereas in this transcript number 2, only 2 of the 3 were spliced together. And so this would be a 66% efficiency. And so you can see based on the efficiency of splicing that it can produce 2 different transcripts from a single gene. And so this can possibly produce 2 different phenotypes, and maybe a way for females and males to produce sort of these 2 different traits. And so in order to see if alternative splicing is one way for males to resolve conflict, I looked at, once again, our polyandry versus menandry comparison. We'd expect that once we evolve polyandry, that these alternative splicing efficiency would also increase. And so once again, PSI is sort of this the efficiency of splicing altogether. So is it 100% versus 66%? And just to sort of summarize, we see that for female brain, male brain, and ovary, there isn't a significant difference when we go from our monagic comparison to our polyandric comparison. However, with these testes biased genes, we see we have a rapid increase in splicing efficiency, and it's significantly different from monagic comparison. So this does indicate that as we go from monadry to kind of a polyandry mating system, that we have this rapid increase in alternative splicing efficiency. And so this may be a major mechanism for resolving conflict for males and likely optimizing fitness. So instead of modifying pre-existing mechanisms, males may just evolve entirely new mechanisms. So instead of competing for similar genes that are important for males and females, males may evolve entirely new genes to deal with this problem. And so in order to test this hypothesis, we looked at seminal fluid proteins that recently evolved in honeybee that were specific to honeybees. And we were looking at differences in evolutionary rate. And so we worked with a collaborator, Rob Waterhouse, to kind of to provide us with some evolutionary rate analysis. And so he looked at evolutionary rate across Hymenoptera. And we were interested in whether seminal fluids had differences in these evolutionary rates. And so we see that when we looked at contraction and duplicity, that seminal fluids had a significantly higher rate of contraction or gene loss, but also a higher rate of gene expansion. And so you can see that some of these newly evolved genes have these differences in evolutionary rates. And this may be a form of conflict resolution. And so just to summarize, the first point I was really looking for was, how does polyandry impact divergence? And I see that polyandry not only drives a rapid evolution and divergence of male reproductive genes, but it also increases evolutionary rate. Second, I looked at how polyandry deals with sexual conflict. So first, I find that polyandry does increase sexual conflict, especially for testes biased genes. Second, I find that while males may sort of overcome this sexual conflict by not only increasing and changing expression ratio between females, but also increasing alternative splicing efficiency to produce two different phenotypes. Third, I find that males can possibly evolve entirely new mechanisms to deal with sexual conflict, and then that involves rapid gene loss and gene expansion to kind of deal with sexual conflict. And with that, I want to thank my supporters, Brock Harper, Amy Dapper, and Robert Waterhouse for all the support, and obviously all my funding partners. And with that, I'll take any questions.
Yeah, thank you very much, Garett. That was a really cool talk. Molecular mechanisms and things like that. Well, there was a question about, did you have a look at geo terms related to the genes you found, but I understand that you haven't had the time to do this yet. Garett, are you there somewhere?
Or can you see me?
Yeah, sorry.
Yeah,
Hi. Yeah, I think you answered already a question about the fact that you haven't had the time yet to have a look at the geo terms enrichments of the genes you have found. But I think I would have a related question. Sometimes you use seminal fluid and sometimes you use testes. Is there any reasons for that?
Yeah. So the one reason I use seminal fluids is because those are kind of sex limited, right? Because they're only expressed in males and not females. And so we have different hypotheses for genes that are sex limited versus sort of sex biased. And right, so we expect these to evolve at sort of different rates. And so I use seminal fluids as sort of my sex limited categories, because they're only expressed in males, whereas the sex bias, the testes biased ones are biased in males in the testes tissues, for example, if that makes sense.
Well, you mean that, yeah, you could also have some housekeeping genes, but the genes, did you select genes based on the fact that the previous study had shown that they were specifically expressed only in testes versus housekeeping genes or things like that? Or is it just you took the whole bunch of genes you found expressed?
Oh yeah. So I did my my bias analysis, and I think that they had like a 2 to 2.5 logfold change between multiple tissues, and I categorize those as like testes bias, for example. And so there's like low level expression for some of them, and I didn't talk about this in the presentation, but I did do like sex bias and then like sex specific or like, tissue specificity analysis. And then I found like more extreme results.
Yeah, because well, remember you used, and is there any specific reason why you chose a brain? There's one I could see eventually that is more genes expressed than any other somatic tissue, or?
It was, yeah.
This is basically the reason. And then finally, on the same sort of things, I was just wondering, seminal fluid, that includes sperm itself, I suppose.
It includes just the seminal fluid. So I got these genes from a proteomic analysis where they separated the seminal fluids from the sperm. And so these are just the seminal fluids. And the ones I included throughout were the ones that were non-orthologs to the other bee species. Because there are some seminal fluids that are orthologs. Yeah.
Okay. But then in terms of competition, I was just wondering, because I can see competition between sperm cells for insemination, but all the seminal fluids from different bees will be all mixed together once insemination is done, or am I wrong somewhere? I'm not a bee expert myself.
Yeah. So there's some really cool competition. So when the queens go on these multiple mating flights, she'll probably mate with like 7 to 15 males, and the sperm has to go through this migration route to the spermatheca, which is sort of the sperm storage organ. And so there can be these different kinds of sperm from multiple males. And what is shown is if you expose sperm from seminal fluids from other males that are maybe non-brothers, that this can decrease viability. So there is probably some competition with how the seminal fluids interact with other sperm groups. Yeah. Yes, especially since there's only like 5% of the sperm actually get accepted. That's post-insemination.
There's a question here. Follow up on the point about geoterms. For many of these rapidly evolving genes and gene families in reproductive tissues, there simply are no good ways to functionally annotate them in any detail. Yeah, because I think I understand many of the genes in Honeybee genome don't have very deep annotation. I mean, sometimes it's just similar to this and that.
Yep, and that's a really good point by Rob. Yeah, they're just no good ways to functionally annotate them in detail. So yeah, that's definitely a good point. And yeah, that's sort of the next step I haven't really approached yet.
Well, especially if you've got gene families expanding and contracting, sometimes you have new functionalization and things like that, and new functions appearing. So it all makes things so much more complicated.
Yep.
Yeah. Okay, so I'll take the next question. So the sperm is already fully formed in the late pupae and then migrates to the seminal vesicle during the adult days. While the tests degrade. What was the age of the gene expression analysis when you compared drones with queens that start to activate ovaries after the mating flight? Yeah, okay. Yeah. Did you have like differences in age of the individuals?
No, that's actually an excellent point. I guess I should clarify this, and it's something that I didn't, clarify during my talk. Is so I'm collecting like sexually mature drones, which were at like day 15, and so yeah, the testes have degraded and the sperm have already migrated. So I guess I did expression analysis on the whole reproductive tract. So it wasn't just testes specific, and yeah, that was kind of error on my part. I should have just said like reproductive tract instead of just testes. So yeah, that's okay. That's true that once they sort of emerge as adults and the sperm starts migrating to the seminal past the testes, and then the testes do degrade. Yeah.
Okay, and well, we already touched a word on this, but perhaps you have something. Did you check for tactic-free, in the expanded genes of for signatures of new or subfunctionalization? Well, this relates to the annotation, I should say. If you check for new functions arising for the duplicated genes.
Yeah, that's definitely one question I didn't really look at. Yeah, I just kind of focused on the seminal fluid proteins to begin with, but that'd be sort of a cool next step.
There's a question also about the link to the genetics. Did you check if some of the genes you find are located close to recombination hotspots or close to centromeres? would be interesting to see if genomic patterns of association between them differ across some mating strategies. Yeah, this is related to recombination and selection is sometimes pretty much linked.
Yeah, that's an excellent question. I do know some from my previous analysis that because I also looked at haploid selection and how that affects recombination rate, and I found that genes that were biased in some of these male testes did have a higher recombination rate, but I didn't specifically look at whether they're located next to hotspots. But I think that'd be kind of a really cool question because this recombination could be driving some of this variation that we see. And then
okay, and there's one last question from, oh no, there's more, but okay, I'm going to go with the question of Brenda. I think you touched upon it a little bit, but she's asking if you identified tissue-specific genes in your data set, or is it based solely on expression?
Yes, it's based on expression. And yeah, so throughout this talk, I kind of focused more on biased genes. I did look at like tissue-specific expressed genes, but I didn't include that in the talk because I was kind of out of time. But I do have 2 different types of expression groups.
Okay, so next talk can be on the other stuff.
Exactly.
On the thing that you didn't show us.
Yeah. There's also a question perhaps related to blood recombination. In fact, to be interested to have some insight on the chromosomal location of the genes and whether there are some patterns. Well, previous question was related to recombination. There could be some other pattern.
Yeah.
Yep, exactly. Well, and so I just kind of elaborate kind of my next steps, I guess, because I might be able to answer some of these questions. So, this is kind of, I'm looking at some of these mechanisms that shape genetic variation, but I guess the next step is kind of looking at kind of the quantitative genetics, right? Trying to identify the genes associated with fitness. And so once I identified genes associated with like sperm viability and sperm concentration, I might be able to answer some more of these questions. And so this is kind of a first step in seeing how polyandry and haploid selection sort of as a mechanism impact genetic variation. And then next I'll be kind of looking at more of a quantitative genetic approach to identify sort of specific genes are associated with some of these traits. And so that's gonna be kind of a next step I'm going into.
I had a technical question. Another question. I had a technical question on the splicing efficiency. What do you mean exactly by splicing efficiency? Is it the exon get removed more and you have genes which a lower number of exons, so smaller proteins, or is it just?
Yeah. So from that example, I guess I can both the talk, I guess, but let's say you had like kind of 3 exon locations and they're all spliced together. That'd be 100%, right? Because all 3 of them are spliced together. Whereas if it's only 1 out of the 3, that's 33%. And so the splicing efficiency would be lower. And so that's one way we looked at it. I forget which program we used, but we looked at kind of splicing efficiency of these regions.
Okay, I would have thought it was the diversity, the number of different variants, splice variants you would have found. It's really, well, it's like shrinkage of the final protein product.
Exactly. Yep. That's a good way to put it. Yep.
Okay. That's interesting. So are there other questions? My Zoom is
Okay, I guess. Do you have one more question, or we can?
Well, we can go on in the chat eventually.
I'm Slack here, so if you have any questions.
Yes, but it's if there's more questions.
So that's this really interesting talk, and I can see you've got still a lot of analysis to do. So we'll have to come back to tell us what you find.
To see the rest.
And I want to see the rest of what's coming up next.
Appreciate it. Yeah, especially.
Okay.
If anyone has more questions, don't hesitate to put them in the Q&A, and Garett is still let you answer them, I think, so that's great. Thank you very much.
And the Slack channel.
And the Slack channel, sorry, yeah.
Okay, so thank you very much, Garett. So now the final talk is by, I'm not sure I will be able to pronounce it correctly, Jana Obšteter, who is a research associate at the Agricultural Institute of Slovenia. She's a quantitative and genome analysis trained in human genetics, University of Edinburgh. She focused in her PhD on developing efficient and sustainable genomic selection strategies for small dairy populations. And now she is focusing on honeybee breeding. I think coming from Slovenia, I understand that honeybees are very important over there. So she will now present a new simulator of honeybee populations designed to test different breeding strategies and design sustainable breeding programs. So we're going now a presentation who's going to make use of genomics for the future of honeybees. So Jana, it's your time.
So hello all, my name is Jana Obšteter and I'm a quantitative geneticist that has recently developed love and passion for honeybees. So I started collaborating with the honeybee team here at the Agriculture Institute of Slovenia, and together with the colleagues from the Brazilian Institute, we've developed a stochastic simulator of honeybee populations and breeding programs that we've simply named SIMplyBee. So I want to start off with a quick introduction about the honeybees, just because we have a broader audience here and also it will be helpful later in the presentation. So the honeybees are insects. They come from the order of Hymenoptera and they are social insects. This means they live in colonies. So at the top, they have this hierarchy. At the top, there's a queen. There's only one queen per colony, and it's also the only reproductive female. Then we have up to 60,000 workers per colony, so they are females that do the tasks but don't reproduce. And then we can have up to several thousand drones per colony, so they are the only males, but they are also reproductive. A substantial genetic peculiarity of the honeybees and also other species from the Hymenoptera order is the haploidiploid system. This means that the queen and the workers, which are female, are diploid and have two sets of chromosomes, whereas the drones are haploid, have only one set, so they develop from unfertilized queen eggs. They also don't have a sex chromosome, but the sex is determined based on a genotype on a single locus, which is called the complementary sex determination locus. If an individual is heterozygous on this locus, it develops into a viable female, but if an individual is homozygous at this locus, and in a diploid state, of course, it develops into a non-viable male. And these males are not non-viable because they have any morphological or physiological issues. It's just that the workers eat them as soon as they realize what's going on, and this is pretty fast. Another reproductive specificity of the honeybees is polyandry. So this is where the queen mates with several drones at once, or with several males. And this is not something that we would usually see in other species, especially mammals. So the drones gather in this drone congregation area, which is a large cloud of drones from several colonies nearby. They gather somewhere in the air, and the queen comes here and mates with several of them. And then the drones die as soon as they mate. The next specificity is the phenotype. So the phenotype is observable on a colony level, not on individual level, which is again not something we are used to in other species, just because they work as a unit. And this phenotype is affected by the queen effect, so how well the queen manages the hive, the colony, and the worker effect, which is how well the workers perform the tasks. And this is usually true for most of the traits that we observe, and of course there's the effect of the environment. And the last thing I want to mention here is the colony events, because the operational unit in honeybees is not an individual, but it's a colony. There's a couple of events that can happen and that can affect the performance of the colony and also the dynamics of the population. So the first is swarming, which is where the colony or the hive just gets too crowded and the old queen leaves with a part of workers. Then the next is supersedure. So this is where the old queen dies or is somehow else removed, and then the new queen takes over. So a new virgin queen is reared from the virgin cells. And then there is split. So this is a managed action where we take part of the bees away to prevent swarming. I also want to mention the genome of Apis mellifera because it's quite a special one. So it's a small genome. It has only 250 million base pairs. on 16 chromosomes, but it has a really high recombination rate. So our recombination is about 100 times more likely to happen than a mutation. This means that the genome sums to 52 morgans, whereas the human genome, for example, is only 30 morgans in length. So it's quite a large genome in the genetic sense. So I will split this presentation in 3 bits. First, I will talk a bit about the simulations in general. Then I will present our package SIMplyBee. And lastly, I want to show you how we've used this package to model a yearly cycle of a colony. So simulators have become quite an essential tool in testing different breeding strategies and methods. And it's just because this has too time-consuming, money-consuming, they're too costly, and they require too many animals to be executed in real life, especially if you're dealing with species that have long generation intervals. And there's quite a lot of simulators out there. They are mainly adapted to simulate the most commercially interesting animal and plant species. There's also some simulators for the honeybees out there, but they model the quantitative genetics bit and the relationships between the bees really well, or they model the colony events and the dynamics within the hive really well. But none of them really cater for a holistic simulation of a honeybee population. So one of such simulators is AlphaSimR, which is an R package for stochastic simulation of breeding programs and populations. And there are some basic steps in this simulation, and this is common to many simulators. So first you need to simulate a genome. This genome has to be representative of your species, which means that ideally it will represent the demographic history and all the genomic patterns of your genome. Once you have the genome or the haplotypes, then you create your base population. So you combine these genomes into individuals of a desired ploidy. Then you can simulate genetic effects, which means that you pick some sites and you sample some effects from them, and then you can compute the genetic and phenotypic values. And once you have your individuals, you can use them as building blocks to combine them or do whatever you want with them to create a breeding program that reflects what you already have in real life or something that you would want to have in real life and you want to test. So at the end, you can compare different measures. So we usually look at the genetic gain, which is the aim of the breeding program, so which strategy performs best. You can look at the genetic variability, so genetic or genic variants, which are important for long-term selection, but also for conservation purposes. And if you have the estimated breeding values, then you can also check the accuracy of the methods that you were using. So AlphaSimR is very flexible, but it doesn't cater for all the peculiarities of the honeybees and doesn't really allow for the simulation of honeybee populations. And that is why we've created this SIMplyBee, which is actually an extension of the AlphaSimR, which means that it holds all the flexibility and all the features of the AlphaSimR. It just adds these bits that allow you to simulate honeybee populations. So the SIMplyBee accounts for the honeybee genome, which really has to be simulated for each species individually because each species experienced a different demographic history. It accounts for the haplodiploid inheritance. It accounts for the caste system, so the fact that there are many castes which are different and perform different tasks. It accounts for the complementary sex determination, for the polyandry, for the colony events, for the direct and indirect genetic effects that contribute to the phenotype, and also for the spatial context. For the spatial context, in a sense that bees are still quite stationary and mate in their vicinity, but also in a spatial context of environment quite heavily affecting bees' performance. So now I'll show you how we've dealt with simulating honeybees and what solutions we came up with. So the basic operational unit in AlphaSimR is a population, so a pop class, that holds individual level information. It holds the ID, the parental information, the sex, and then if you've simulated traits, it also holds the genetic value for each individual, as well as the phenotypic value, and it can potentially also hold the estimated breeding value, and there's a miscellaneous slot. So just to emphasize, the genome and the genotypic values are simulated for each individual, so for each bee. But because for honeybees, the population is not really the basic operational unit, but instead it is a colony, we've created a colony class. And this now holds colony information. So it holds the ID and the location, and then holds all the casts as separate populations of individuals. So each of these populations looks like the one on the previous slide and holds individual level information. Then we have a phenotype slot, which is just one value that represents a combined genotype, phenotype of the colony. So it's a function of the individual phenotypes plus the environment. Then we have a couple of slots that tell us what happened to the colony, which is quite important from a perspective of management, but also performance. So this tells us whether the colony has split, swarmed, or superseded, and these events can be reset at any point. Then we have a slot that's called production, and this is production in the sense of a trait that we're simulating. So because honeybees operate as a colony, and phenotype is expressed on a colony level, it might happen that if the colony is too big, you might not have a phenotype to observe. And this is what this production slot will tell you, so whether the phenotype is actually observable or not. And again, a miscellaneous slot. And because beekeepers and also honeybee researchers usually don't work with specific colonies, but work with apiaries or even populations, we've created a colonies class that holds many colony objects. So it performs like it acts like a list of colonies, but has additional functionality. So according to this, we've developed a bunch of functions that we've split into 3 levels. So level 1 will work on and return a population class. Level 2 will return a colony class, so it allows you to perform some functions directly on a colony. And level 3 will work and return a colonies class, which means that a function will be applied to all the colonies in this object. So an example of level 1 functions that will return a population is, for example, a function that will create a queen, worker drones of virgin queens, so individual populations of casts, a function that will get or pull some individuals from these casts. There's a function that will create a drone congregation area from the list of colonies that you supply. So this will simulate the cloud of drones over in the air where the queen comes to mate. There's a function that will pull drone packages from the DCA, a desired number, and this basically samples the drones that the queen will mate with and then also removes them from the DCA because they die. And there's a function to cross a virgin queen and so on. The level 2 functions will return a colony object. So there's a function to create a colony, either empty with a mated or a virgin queen, and a function to cross the colony. So what this does is it crosses the queen, but then also adds in workers and drones. We have functions to add, replace, remove any individuals of the casts in the colony. We have a function to requeen the colony. And then we have a set of functions that are quite important because they simulate the dynamics within the colony. So you can simulate that the colony swarms, that we've split it, or that it supersedes. And this, again, affects the performance, but also genetic variability in the population. And then there is a function that will set the phenotype to the colony, so it will be a function of the individual level phenotypes. And this is an example of how you would use this level 1 and 2 functions. So you would start by creating a base population alpha sim R. You would select a single individual to be a queen and several of them to be drone producing queens. Then you can create the drones and create this DCA from the drone producing queens, and let's say you want 100 drones per queen. Then you can pull drone groups from the DCA, so pull the drones that the queen will mate with, so the fathers basically, and here we want we want only one package with an average group size of 15, and this is sampled from a truncated poisson. And then we have the queen, we have the fathers, and we can create a colony. So we provide the queen and the fathers, and in addition, we want this function to add in 100 workers and 10 drones. So these are demo numbers. Once we have the colony, we can do whatever we want with it. In this case, I will swarm it with a percentage of 0.5, which means that 50% of workers will leave and 50 will stay. So this returns two colonies. One is the swarm that leaves with the old queen, and one is their remnant. And I can do whatever I want with these two colonies, either assign them back to the original one or create a new one. And lastly, we have the level 3 functions, which work in the same way as level 2, just instead of applying it on a colony, it applies it on the list of colonies. So there's of course a function that will create this colonies object, either with empty colonies, mated or virgin ones. That there's our functions that will add or select some colony objects from this colonies list. There's a function that can build up all the colonies in this list. There's a function that can cross all the colonies to a certain DCA. There are functions that will split, swarm, supersede, or collapse all the colonies in this list of colony objects. And this is an example of how you would use these level 3 functions. So first, again, you create a base population and sample some drone-producing queens from it. So the queens that will provide drones for your queen to mate with. So then you use function create colonies to create an apiary, which will now have 5 colonies in, and they will all be virgin. So mated will be set to false. Then again, we use function create drones to create this DCA from the drone-producing queens. Again, but now we'll have 1,000 drones per queen. And then we will cross all the colonies in the apiary. So all of the virgin queens will fly to this DCA and mate. And once we have them mated, we can build them all up to the, let's say, full size, but here I'm building them up to 1,000 workers and 20 drones. Again, these are just demo numbers. So in the package, we've also dealt with simulating the CSD locus. So we specified a region on the genome to represent the CSD, which is a sequence of non-recombining biallelic SNPs. So the length of this locus, or the number of the sites in this sequence, then determines the number of CSD alleles in the population. If the CSD is turned on, it can also be turned off, then all the functions that create individuals will remove the homozygous brood, but will save the percentage of what was removed. So you will know how many of them were non-viable. And then we have functions to extract either the number of distinct CSD alleles in a population or colony, or to extract the alleles directly. So lastly, I will show you how we've used these functions to simulate a yearly cycle. And we've modeled a yearly cycle of a managed apiary. We did 20 or 40 colonies and 20,000 workers and 4,000 drones per colony. And in this population, we had 128 possible CSD alleles. So we've simulated a yearly cycle based on what actually happens in a honeybee colony. So we've started by creating some base population and some mated colonies. So we had to start somewhere. But then we've entered spring and simulated what happens there. So the first thing in spring, all the colonies build up so that they have enough individuals to perform all the tasks they need to. Then, because this was a managed apiary, we split and requeened all the colonies to prevent swarming. But then still, a small percentage of them still swarmed and a small percentage of them still superseded, and we've selected them at random. And then we've also collapsed a small percentage of the colonies just because some of them also die in spring. Then we enter summer. So in summer, the colonies don't build up again, but a small percentage of them will still swarm, and a small percentage of them will still supersede, and some of them will still collapse. And this is also the point where you would collect phenotypes, but we didn't do that in our simulation. And And then comes the winter, but nothing much happens, but the colonies do collapse. So a larger proportion of colonies collapse in the winter due to overwintering issues, but also diseases. So what we did here is we sold the splits to keep a constant size of the apiary and collapse the larger proportion of them. So we've repeated this for 10 years and in 10 replicates, and we've inspected how many of the CSD alleles were left after 10 years of this closed mating scenario. So here I'm showing a number of distinct CSD alleles in either an apiary or a colony, when we had 20 colonies in an apiary in green, and in blue where we had 40 colonies. And again, this was a closed mating scenario. When we have 20 colonies, an apiary samples about 120 CSD alleles, but in these 10 years of closed mating, it loses nearly half of them. The situation is a bit better when we have 40 colonies, so we lose about a third of them over the period of 10 years. The dynamics within the colony is a bit different. So every colony samples about 25 to 30 CSD alleles. But once they start mating, this increases, and then the inbreeding kicks in, and this drops below what it was originally. So this is just a very simple example of how you would use this simulator. Because this simulates an individual level beads. So at the end, you could extract anything you want from it, any measure, based on individual or colony level. So future work, the first big step will be the simulation of the phenotypes. So like I mentioned, both queen and worker effects affect the phenotype in kind of honeybee colony. So at the moment, it is possible to simulate a colony phenotype, but the user has to specify a function of how to combine individual phenotypes into the colony phenotype. And what we want to do next is we want to work on good defaults for this that would account for everything we know about quantitative genetics of the honeybees. And the next big thing is to simulate the honeybee genome. So currently, this is still not implemented. We've added the honeybee to the STD-PubSim, which is an online, freely available catalog of species information and also demographic models that allows for standard simulation of genomes across studies. However, we only added the genome information, but not yet the demography. So what we are working on now is to inferring and building a good demographic model of the honeybee history that would allow us to then simulate a founder population, so an accurate representation of the honeybee genome by subspecies. So lastly, I just wanted to mention that honeybee is under active development, and it will shortly be released as an open source for community to use and extend, so for all of you. But if you had any ideas, some comments, something that you would like to suggest to us, or maybe something that we could do better, please let us know, and we will gladly take any advice or comment. So this is all from me, and I'm looking forward to the questions. Thank you.
Thank you very much, Jana, for this really nice presentation. Maybe you can unmute yourself if you're around and put your video on. I don't know, is Jana around?
Yeah, I'm here.
I don't know. I was worried because you said you had some trouble to connect earlier on.
Yeah, I don't know whether my video is working or not, hopefully.
I can't see you right now, but at least we can hear you. So I guess that's already good. Yeah, here it comes.
Okay.
Okay, so thank you very much for the really nice presentation and for the tool you're developing. I have some questions about the phenotypes. So I know it's some work in progress that you will be adding to the development of your program, I guess. But I was wondering which type of phenotypes you wanted to include and how you model them, because we have some phenotypes that we can measure on individuals, some that we can only measure on colonies. How are you taking that differences into consideration, and which type of phenotypes you are thinking of including in the model?
Yeah, so just a second, let me just see whether I can get my camera on, or maybe something has gone wrong, no idea. So yeah, the phenotypes are quite interesting in honeybees, because we observe them on the colony level, right? But they are still expressed on an individual level as well. Sort of honey yield is still expressed on an individual bee level. It's just that what we observe is the colony level phenotype. So with this simulation, it would really allow us to simulate the individual bee phenotype, but then the question is, yes, how to combine them. So all the methods that dealt with that up to now have sort of modeled the phenotype as a queen plus a average worker effect, but now we will have individual bee effects. So we have to somehow combine them with a covariance between them in mind. So that's a work in progress. Regarding the types of phenotype that we would simulate, well, it's up to the user as long as they specify the right variance components or the right correlations between the traits. Is really up to them. It will, and like I said, it's observable on individual and the colony level. But what we were coming from is, of course, we started building this because we want to find good solutions for our own breeding programs. So we are collaborating with Ross, and they are creating a breeding program for the UK, and we are creating one for the Slovenian conditions. And there are really no good tools that we could test that up front. So the phenotypes that we would have in mind are, of course, honey yields, gentleness, the things that we are selecting on. But also here in Slovenia, we are in our breeding program, we are selecting on the color of the bees, which might be a great disadvantage. And we could be throwing a lot of the genetic gain away just because the bees are not of the right color, and we don't actually even know what is causing the color, whether that's purely genetic or that's maybe environmental things involved in that as well. So that is also a part of the reason why we're building this, and also want to test what actually would happen if we wouldn't be doing this.
Okay.
I was just wondering about phenotypes. In a colony, we have patchy lines. I mean, workers can be either 3/4 related or just 1/4. Would it be possible to have some phenotypes which would be influenced by the patchy line, and when your colony grows, according to, I mean, the number of workers from one patchy line would sort of keep that sort of phenotype, and perhaps also you could have different growth rates for different patchy lines or things like that envisaged?
The evolution,
yeah.
the number of patchy lines.
Yeah, so that's a good point for us to consider. Thank you. Regarding the genetics of the patchy lines, well, because we are simulating individual drones, each patchy line will have a different genetics and will have a different phenotype. So I think that will be covered. Regarding the growth of the different patchy lines, yeah, that's a good point, and I think we'll have to brainstorm about that. Yeah.
Yeah, because I think we can imagine that the number and the proportion of each patchy line making the colony will evolve through time. And yeah, maybe also like I had like some, I don't know if it's question or suggestion really, about taking into consideration the yearly dynamic of the colony in a specific environment. For example, if you have kind of like blocking nectar flow that would stop the brood from growing at some point. I mean, can you include this type of variations in your simulations and through time, and maybe also involve like some dynamic of the pathogen, for example, varroa or things like that?
Yeah, so we want to make the size of the colony as a function of something. We don't know what yet, something genetics and environment. So it will have to be a function of that, and that will be the next thing we have to implement. Currently, the user has to modify it with the event, but we want to make that a function of genetics and environment. So hopefully we will come to some good solution for that. But of course, we can't really automate everything. Something will still be up to the user. But as long as we provide flexible solutions so that the user can input maybe some functions because they change according to where you are. I think it could cater a lot of different
that's
environments.
what I was thinking, like if you can, for example, provide different distribution of your dynamic as an input to the model, that could help the simulation to fit the specific environment you're in or you want to test.
There's a question. Well, Jay Evans, a great talk, and simply looks like a really nice practical and learning tool. Yeah, you can learn things using this, I suppose, how colonies work and things like that. Can you layer in more environmental factors that might affect genetic by environment traits, thinking of whether a beekeeper controls mites or not, or local pesticides, insults that some genotypes handle better than other, winter length, etc. Not knowing the modeling platform so well, how flexible can that be? I think it's
So I managed to turn on my camera.
We have you.
Hello to you all. It's really difficult in bees because just the sole phenotype or the size of the colony is a function of so many things. But maybe, like I said, we are open to any suggestions you might have. But for now, I'm guessing you could simulate multiple traits. One could be the resistance to varroa. One could be susceptibility to pesticide. One could be overwintering ability. And then you will have to somehow model a combination of this phenotype. So we could come up with some functions that would gather them all and help you model the strength or the phenotype, something like that.
There's a question of Martin Hasselbalch regarding the CSD allele. You use length variation only. This would ignore variation in composition of alleles at the same length that are heterozygous too. Any chance to implement this?
Composition of CSD alleles. So no, it's not just the length. It's the length of it's a sequence of SNPs. There are biallelic SNPs, and then the actual sequence of this of the alleles on the slow side then is the CSD allele. For now, I said length because if you say, oh, my CSD allele is only 2 base pairs in length, well, then you have only 2 possible combinations or 4 possible combinations. But then if you say, oh, my CSD allele is let's say 5 base pairs, then you have 32 possible combinations, and that is excluding mutations for now. So there is we are simulating the real alleles, but this that I've shown we didn't simulate mutation, which of course happens on the CSD alleles, but this was just like to play around with the tool and to get a good sense of whether it's working or not.
I suppose it's a sequence of 1s and 0s which can model either SNPs or insertions and deletions just the same way. I mean, when it gets into the simulator, it doesn't see the difference, I suppose.
Yeah, exactly. It's just if the 2 alleles are the same or not.
Okay. Well, regarding the CSD, I had a question. At one moment, when you explained the tool, you mentioned using 20 drones from the drone congregation area. Well, did you keep the same setting for the example you showed in CSD, or did you allow more drones? Because I saw that the number of CSD allele went up to 35 or 40, so within a colony.
So that code that I've shown, that was just a toy code for you to see how the tool works. When we did the simulation, we actually, because we simulated one apiary, so the queen was allowed to mate with all the drones that were currently present. So that was, if we had 20 colonies and about 4,000 drones per colony, so all of those drones were in the DCA.
Yes, but I mean, she only draws a subset of these drones. It doesn't get actually mated by the whole thousands.
No. So we create the DCA like you would see it in real life. So we gather all the drones, but then we sample only 15 of them for queen to mate with. But that's a random process of selection, which is maybe what would happen in real life.
And you also mentioned modeling the genome as it is. Can you include genetic maps which would reflect a higher or lower recombination? Well, you do have an example with CSD where you suppress recombination. So I suppose there could be a mechanism of doing it for all the markers, having rates.
Yeah, of course. So how we are currently dealing with it. So we have implemented the simulation of the real honeybee genome according to the parameters. I don't think we've included the genetic map at the moment, but we certainly can. The simulator allows us to set any genetic map we want. But the biggest problem with the genome we currently have is that we can't simulate it with a good demographic model. And Alice explained how we know where the bees came from, but we don't. What we are lacking for the simulation is actually the detailed parameters, so the effective population size and the time of these events, and they are so uncertain. And also the effective population size was estimated only in 1 or 2 studies, so that is the thing we are lacking for the accurate simulation of the genome. Yeah.
Yes, because you included mutation rates also. How are these estimated? Yeah.
Sorry?
No, you included also mutation rates. Are these really well known for bacteria or?
So I think there's only one estimation of the mutation rate in honeybees, and we are relying on that one. We have no other choice. So we're doing the best we can with what we have at the moment. Yeah.
Okay. So this means we still have lots of work to help speed up this sort of software. So, well, I have some more questions that I'll go in the chat because it's getting 7 o'clock over here, and I think you have to leave also. So,
thank you very much for your presentation and for answering all these questions.
Yeah.
I guess we can say a small closing word. So thanks everyone, like in the organizing committee. Everyone that is behind the screen, Glenn, Brad, Pia, Anna, Lindsay. Thanks to all the presenters. That was really nice to see your talk tonight, and I'm really looking forward to see more of your work in the future because it was really interesting and fascinating. Thanks to everyone that joined us tonight and that was here to listen to these talks, and you can find them online in the coming weeks on the YouTube channel of I5K. The Slack is still open if there's questions that people that missed the presentation have, or I hope the presenters will make themselves available to answer them on the Slack. And I guess with that, I have to say thank you to everyone. Have a nice evening or morning or day, depending where you are.
Thank you very much.
Okay, thank you.
Goodbye. See you next year in real life.
Hopefully.
Hopefully.
Bye.
Bye.
Thank you. Bye.